I just wanted to trigger some discussion or stimulate some thoughts about new characteristics of mutational signatures that can be discovered through experimental modeling. As we have seen yesterday, there is a relatively fixed number of signatures now, but still among those, there's about 35% orphan signatures of unknown etiology. And also there's a question if we are still missing some signatures in the pan cancer data that just wouldn't pan out from the typical bottom-down analyses such as NMF. And the rationale for doing experimental signature modeling is basically listed here. We can validate the known signatures identified in tumors. We can identify the causal factors underlying the orphan signatures. Perhaps we can discover these yet unrecognized signatures of cancer risk factors. And then we see some special features such as in composite signatures, we can basically see different contributions of different factors. And finally, in certain systems, we can also identify the functional impact of signature of mutations supporting the signatures. This is just to give the overview of the integrated experimental system and how it's compared to the human sample analyses and mining of public data. And my team basically operates mostly at this level, looking at in vitro systems, some animal studies as well, and occasionally we do human samples, and we certainly mine the cancer data. This is not an exhaustive overview of experimental systems. Papers were also published on yeast, C. elegans, as you know, and so on. Okay, we use clonal expansion assays, which could be either based on primary cells, and one well-established system is based on Hupke mouse embryonic fibroblasts. This is basically a human p53 knock-in modification of the cells and of the animals. And this system in culture goes through senescence, and then it bypasses the crisis stage, and the cells emerge as immortalized clones. And you can expose the cells here and then collect the final clones basically and compare the untreated versus the clonal expansion and to read the spectra. Another system based on primary cells that we used recently is based on human mammary epithelial cells, and this is a little bit more complicated system that we obtained from Martha Stanford at Berkeley. We have done one exposure, but it has been very informative, and I will talk about this a little bit later. But basically, the cells, again, have to overcome certain biological barriers to become immortalized and clonal. And again, you can sequence these clones and compare them to the baseline population. And we also adopted the approach that are now just listed, which is basically using different immortalized cell lines. Chronic sustained exposure for a couple of months, single cell subcloning. Sometimes we are not entirely successful with single cells, so we sometimes see 2 to 3 populations also, but ultimately we do get mutagenized clones from which we could read signatures. The animal system that we are using comes from the National Toxicology Program at the NIEHS. And we are tapping into 2-year chronic toxicity studies, carcinogenicity studies. There's about 600 something of them. They are carried out in mice and rats, isogenic animals, males, females, multiple doses, 50 animals per experimental group. So these are basically huge, very well-controlled studies. And we work with the FFP samples from those animals, tumors and normal tissues. And this is quite challenging because you end up with a very poor quality DNA, but occasionally we do get libraries, they are sequenceable, and they actually provide information. Okay, so I wanted to give you a few examples of what we see in some of these experimental signatures. Most recently, we focused on acrylamide because that's a compound which has been probably human carcinogen. It's carcinogenic in rodents for sure. it forms in heated starch in other foods due to the Maillard reactions. And you can actually find it in foods like, fries, toast, also in coffee, and certainly in tobacco smoke. And acrylamide is metabolized in the body to glyceramide, and that is the reactive epoxide that can attack DNA and possibly other molecules. And here we looked at, we used the MEF system, clonal expansion MEF system, to model signatures of acrylamide and glyceramide. We are currently progressing through NTP lung tumors, and we also are preparing to sequence renal cancers where we have the information of exposure of the patients to dietary acrylamide. Using the MEF systems, we were actually able to define a pattern which is a signature of glyceramide. Acrylamide didn't give us much due to some metabolic limitations of the system, but glyceramide, the metabolic product, actually gave us something that we found very unique because it has 3 major mutation types that are supported by strand bias, and this is, to our knowledge, maybe the only signature that has strand bias on 3 different mutation types. And similarly to what David mentioned yesterday, we also speculated that that pattern could be basically the subcomponent of signature 4. And we managed to support the mutation types by adduct analysis. We were using the LC tandem MS analysis with Fred Beeland. So these are actually formed through adducts that are well characterized. And our question was, is this pattern here, the prominent T2A strand bias peak, contributing to the SBS4 here? And for that, we needed a clean signature of benzoate pyrene, which would be primarily forming the C2A mutations here, and we generated it using the primary human systems. And you can see it basically has no T2A, and this is consistent to what David also described in the iPS cell system. And then we were trying to do, in collaboration with Steve's group, reconstructions of the experimental signatures. So basically, I will just focus on the middle column here. The combination of the experimental glyceramide and BAP signatures basically reconstructs fully SBS4 in a very nice linear fashion in smokers, lung adenocarcinoma squamous cell, in some livers, and also head and neck cancers. And then we found in the PEACOCK data quite a number of tumors where the glyceramide was present, and we actually had to look for it. It would not pan out by NMF analysis, but we did MSIC-ACT approach with the strand-biased version 192 channel version of the signature, and you can see the proportions of tumors, liver, kidney, and additional ones, lung again, and so on. So we basically see a relatively broad, widespread presence of the glyceramide pattern in different tumors. This said, that presence is relatively low, so these are the assignment proportions within the total mutation loads per sample, and they're basically around 10% mostly, 10 to 15% perhaps. So it's not a very pronounced signature in the human data. Now, so we were actually able to show that these 2 signatures of experimental GA, BAP reconstruct SBS4, and under certain conditions of analysis, when we mixed the experimental BAP with TCGA data for lung cancers of smokers with SBS4 or without SBS4, we were actually able to also isolate a pattern which suggested that the SBS4 can be decomposed. So in other way, basically I can show it like this. So this is the SBS4, and after that analysis, you can see that there is a strong enrichment of the BAP-like signature, but it doesn't have that T2A anymore. And then this is maybe not the cleanest separation, but you can see that the T2A separates from the C2A. So the usually very stable SBS4 actually can be decomposed by adding a very strongly defined experimental BAP signature, BAP-only signature. Okay, so to summarize this, we have a new signature of glyceramide then we can decompose the signature of coming from complex sources such as tobacco smoking using the experimental signatures. And there is this caveat that the T2A mutation types can still come from additional compounds, as has been discussed yesterday, DBP, DBPDE, and also Alan Balmain showed that urethane has that very prominent T2A signal in lung cancer induced by urethane. So we still don't know if it's really glyceramide, and we would need to do some adduct type of analysis really to resolve this. I wanted to mention something about some new features of alkylating agents. This was already briefly mentioned yesterday, so just very quickly, we have a signature of MNNG, which is a research compound in MEFs. It looks like that. It matches the cosmic signature 11 quite well. And from Alan's work, there is a mouse version of signature of MNU. And we extended this to whole genome sequenced MEFs. And we actually recapitulated the C2T signal, which is very typical. But we also noticed that there is a lot of mutations still hiding here that are overshadowed by the massive C2T. And they have a relatively decent allele frequency distribution, so we believe that they're basically part of that exposure pattern. And when we removed the C2T just to see the details, what emerged was a pattern which matches very well the temozolomide pattern seen in the iPS cells in Jill Q-Cap's paper and Davis' paper. And also, Ludmil shared some data on secondary cancers after temozolomide treatment, which again recapitulates this pattern. So the alkylating, methylating agents can actually induce either C2T or T2C, depending on the cell context, maybe some differential repair capacity, and certainly this merits more work. In collaboration with Silvia Balbo, we also looked at adducts forming after MNU treatment of the MEF cells, and we actually could identify DA, deoxyadenine adducts, which support the formation of the T2C mutations. Okay. Another interesting features coming from these experimental studies is coming from the work that we did with Steve on aflatoxin B1. Very briefly, when we compare experimentally induced pattern, it is always different from the pattern. It's consistently different from the pattern seen in humans that had all the biomarkers of aflatoxin exposure, HBV infection, P53 hotspot mutation, and so on. And furthermore, you can see that even the experimental systems actually vary. So the two cell lines that we used basically always separate based on the spectrum, and you can see it here in more detail. So there's always something slightly different for each particular system that you use in this case. So can we actually say what is the canonical aflatoxin signature? Maybe not as clearly still. And finally, ochratoxin A is it's a rodent carcinogen that had been hypothesized to be also human carcinogen, but there are no data on that. We decided to look into the effects of ochratoxin A using the MEF system, and surprisingly, we obtained some patterns, but they were not different from untreated, spontaneously immortalized clones. And so we started looking a little bit more into the signatures there. We identified signature 17, which forms as a background in these cells, and also signature 18. And they are both suggestive of oxidative stress, probably coming from the culture. Nevertheless, we were also able to look at the differences between the treated and untreated cells using a mathematical modeling of oxidization potential. This was done in collaboration with Nouri Alpey-Tamis in Minneapolis. And we see a slightly increased oxidation damage upon treatment. And also the treated cells show earlier onset on allele frequency of the signature 17, signature 18 patterns. So we actually believe that even if you don't see overt differences between the patterns, we see a little bit more of the event-supporting effects of oxidative damage by OTA treatment. And this essentially fits nicely with a schematic that has been put together by Ben Schuster-Beckler at Oxford. And this is just to show that signature 17, signature 18 are basically interconnected, either by, if you start with the oxidation of guanine on DNA, you end up with signature 18, but if you start with oxidation of free nucleotide pool, you can actually end up with 17. So there is some kind of interconnectedness between these two signatures. That still doesn't explain really what is happening in the cancers which have high loads of 17. For example, the esophageal adenocarcinoma and gastric adenocarcinomas. And finally, I wanted to mention that some of these systems can also be used to identify cancer driver events. So a couple of years back, we published a paper where we took all our clones treated with different agents, which you can see here. And then we overlaid this for recurrent mutations with cancer gene census panel, essentially, and also some curated epidriver lists. And you can see a lot of recurrence. The red dots are treatment-specific mutations. The black ones are basically not treatment-specific. And this is a P53. We see hotspots in genes such as KRAS, HRAS, mutual exclusivity for complexes such as BAF complex, and that mimics the TCGA data for BAF complex in humans. And also we see some effects, functional effects on cell survival when we manipulate some of these pathways. Okay, and really final note is that what is the next priority for testing? What compounds, should we focus on? We do have a roadmap that we currently go through. There's a new classification of carcinogens based on 10 key characteristics. These are genotoxic, there's ROS, these are non-genotoxic ones, so we try to also select the compounds based on that. And finally, there is a new publication from advisory group for IARC on recommending priorities for the evaluations by Mutographs program here. And this publication basically lists compounds and factors that could be of interest, to these experimental studies. And maybe around this table, we could also see if we can coordinate a little bit better across the individual groups participating in the Mutographs project. And that would ultimately refine this map, which shows known causes of human cancers by organ site due to external factors. Okay, I will skip the conclusions. I would just like to acknowledge the current and former members of my team, but also other collaborators at IARC, many external collaborators and funders. Thank you very much for your attention.